Anything that you want to share that okay, uh, after that we want to do like this or something. What is your future plan? Out of you, how many are already PhDs? Okay, coordinator madam. So would you please share the second this thing that I have shared already yesterday? One, there are two study. One study you have already shared. The second study, would you please share, madam, on screen so that it is no more black screen. Okay. So what happens? There are multiple different aspects to infection control. Okay. So give you a brief idea that what exactly happens in infection control. All these usual things has been tested over the time, and these will give good results as well. But the problem with all the conventional therapy is I'm talking about pathogen control. Okay. So the usual this thing of the conventional therapy is that after a certain point of time, whatever kind of compound you derive or synthesize, it will by and large will become this thing. What do you call this resistant? Okay. Acha, out of you, can anybody tell me whether physical things will have any kind of resistance? I give you one example. In microbiology, there is a process called sterilization. Okay. Now, in sterilization, what exactly happens? We don't want we don't use any chemotherapy or some compound, nothing. What we do is we simply put all our infected utensils inside a pressure cooker and stay there for like approximately 45 minutes at about 120 degree centigrade temperature with approximately 15 pound pressure. Okay, so what is the end result? Everything gets eradicated, including the resistant spores. So there are some pathogens which are spore forming bacteria. Okay, so all of you must be knowing already, spore forming bacteria are those which have a very tremendous this thing capacity to resist any kind of what you call challenging atmosphere. What they do exactly is they'll hide themselves into a spore, and the spore will once again germinate when the favorable atmosphere is there. So even these spores also get eliminated by physical this thing inhibition methods. So what does it mean? It means that if physical methods are very this thing solid, when there is no resistance, nothing. So today, tomorrow, if you want to devise something which is purely something which is not going to become any kind of resistant, there should be some physical attribute to it. Are you getting my point? If you use an antibiotic, you are not using anything physical. It's all chemical. It will dissolve in the water, then do some kind of molecular level inhibition. Until and unless you devise something which will have a physical barrier or physically impact the organism to kill it, it is of no use. So what are the distinct solutions you have? You have nanoparticles, you have composite materials, and all such things. These are some physical thing which kills the bacteria without any possibility to even get resistant any time sooner. Okay, it will take like 1 million years to become resistant to a nanoparticle, but it becomes hardly 10 years to become resistant to a particular antibiotic. So this particular paper is deals with that. Madam, ये कीजिए, इधर उधर कीजिए ताकि फिगर दिखे. The figures are there, no? Do you show the figures? So here also what we did is we used computer simulation at the same time. Full size figures we cannot see, right? Anyway, So we use the computational tools at the same time wet lab methods also. Okay. If you elaborate, madam, you please share the link with them. So what exactly happens? It is only that this is a good method to make your impact in a published form if you have both the computational simulation as well as wet lab. Now coming back to this, madam, yesterday some tools were left we could not able to discuss. So I would like you to share that tool, which one we have not discussed. I think one is left, and rest of the tools I'll share right away here. Not exactly, madam. I have shared some tool, I guess. Just hold on. Have I not? Just one second. I'll just check it out. Uh, just hold on. I want to see what are the links I have shared with you. Just hold on. Okay. Okay. Fine, fine.
Is it the cubic.org?
What, madam? What did you say? I did not get.
The cubic.org that we were working yesterday on.
Oh, we have already done it, right?
Yeah, we have done it yesterday.
Oh, fine, fine. Shagni, you share the tool that I have, the first tool. Share it.
Okay, sir. Just a few minutes.
Hmm. So you need to devise something which is having a physical attribute to it. Okay, you will see in coming years that there is huge scope in pathogen control by composite materials as well as this what do you call nanoparticles. So nanoparticles are already in use, but there are some limitations. The limitations is standardization of the size. So there are many people are speaking many things, and therefore the standardization has not yet been done. So I give you what exactly is the standardization. If you want to design a antibiotic, there are some standard protocols to it. Okay, initially you will do some fermentation, then you will do an HPLC, purify the product, and then you will do some additions and permutation combinations, like addition of alkyl group or a methyl group addition or removal of a nitrate residue or things like that. So these are standard procedure. Every lab will do the same. Are you getting? But no, in nanoparticle, the problem is if every other lab is doing it differently. Somebody will use some organic compound, somebody will use some protein. So we are interested in proteins. So what exactly happens is there is no regulatory framework. Like US FDA is there, no, for antibiotics. So this is the protocol, you have to follow that only. Okay, I give you an example. Please listen carefully. Consider that This we have already done, close it. Antibiofilm we have already done, close it. Toxin spread we have not done. Just hold on. So what exactly happens? So for example, there are many pharma companies, right? I give you one example like this GlaxoSmithKline. So GlaxoSmithKline is producing one antibiotic. It is their R&D product. R&D product means they are the one for the first time in the world they have designed one antibiotic and then did every research and then finally made into a product and put it into the market. So when it comes to the market, no, it is open for all. Okay, what you do, you take that molecule and do infrared analysis. You will come to know what are the what are the functional group present in it, and then you can design also. So once you design, no, you have to put it again for US FDA certification. Now US FDA will see whether you have made it correctly or not. If they find that you have not made it correctly, they will say that no, I want that exact thing. You cannot do any kind of deviation. Okay, so what happens is, and process of alkylation is for every lab it is the same. The process of nitration for every lab it is the same. The process of cleaving a protein or digestion of a protein is same for every lab. So, but for nanoparticle, no, if you read out researches throughout the globe, you will see that every other lab is using every other method. Getting, so this is a regulatory problem, not a R&D problem. Anyway, now coming back to this, so this is a toxin spread server. If you can please read out, we use random forest algorithm here. Okay, today we will be talking about two algorithms. Random forest is one, the second one is hidden Markov model. So both are supervised machine learning algorithms. So you can ask me, sir, why you are not using unsupervised? Because we are not using unsupervised because we are confirmed that which kind of result we want. We are having a proper what you call is a training set. I'm repeating it one more time. So you may ask me, sir, why you are not using any unsupervised algorithm? Every time either you say support vector machine, artificial neural network, now random forest, and then one more is hidden Markov model. So all these things, no, if you record this, you'll and later on check it out in other books, you will find that these are all what do you call as supervised algorithm. So we are very much sure which kind of output we want. Okay, it is not that we are searching something in the haystack. So this particular tool will give you an idea that what are the different proteins which can cause some toxicity. So I would like to, because this is the last day, no, please somebody tell me what do you mean by a toxic protein. So what could be a toxin? In a protein, what could be a toxin? Can anyone give me an idea of a toxin which is also a protein? Please write it down in the chat box. Any toxin, any example if you can give. It could be a microbial toxin also. Can anybody tell me what is a toxin? Which kind of this thing is a toxin? So there should be some justification. No, why? See here, select machine learning algorithm. So you can use a random forest, then blast, then MRCA1, or you can also use only the random forest. Select the threshold value. Threshold value usually 6 and 5. These are okay. Do not go for extreme. So what would happen is you will lose many potential candidates. No, Shayak Mondal, please listen. I am asking. Anyway, Shayak Mondal has given it a try. I am thankful. At least it has become a little interactive. Please listen. What I am asking here is, can any one of you give me an example of a protein which is toxic? Okay, proteins are like, as you know, this could be an enzyme, this could be a functional protein. Lead is a toxin, that is fine, but it's a metal. So it's a heavy metal. So I'm not talking about that. I'm talking about some proteins which could be toxic. No, that is also not correct. GABA is gamma aminobutyric acid, which is basically a neurotransmitter kind of thing. Okay, so I don't consider it as a toxin. I give you one example, then everybody will agree. Okay, acha. Have you heard of this thing? Alpha amylase? No, no, no. Mary, alpha amylase is a enzyme. That you are right, but it is not a toxin. So what is a toxin? A toxin is something which will cause some damage to you. Alpha amylase correctly is an enzyme. That is absolutely correct. You are right. But in this particular tool, no, if you see the name of the tool, it is toxin pred. So it will help us to identify if a protein is toxin for you. Are you getting? Okay. Uh, so what are toxins? There are multiple proteins which are toxins. Okay, I'll give you one example. Then everybody will say, "Oh, we knew it. I could not tell." So there are multiple diseases, isn't it? Have you heard of cholera? Who can tell me whether they have heard of cholera? Just 1 second. What you have written? Just hold on. Any pore form? That is right. Antimicrobial peptides are the pore forming perforins. Yes, correct. Very good. Yes, you are there. That is a very good answer. But what? No, I want some gross level this thing. Botulinum is correct. Absolutely correct, Shaikh Mondal. You have given a very good this thing, but there is a problem here. Clostridium botulinum is a spore forming bacteria. Diphtheria toxin is the thing I wanted. Akanksha Mishra, thumbs up to you. This is the thing I wanted very badly. Anyway, come back. Why I was particular about this particular terminology? I'll just come back. So one by one. So botulinum, Clostridium botulinum is a spore forming bacteria, gram negative spore forming bacteria, very dangerous. And then this is facultative anaerobe also. Facultative anaerobe, you know, very dangerous. When it can remain inside oxygen, outside oxygen also. So coming back. So Clostridium botulinum produces a toxin, botulinum toxin, and but that is not a protein. It's a secondary metabolite. Are you getting my point? So it's a secondary metabolite, something like its basic nature is like something like a antibiotic. Are you getting like Streptomyces griseus? It produces streptomycin. So it's somehow like this chemotherapeutic, organic substance, but not a protein. Are you getting my point, Shayak Mondo? So botulinum will be a bad example for a protein. But this thing, no, diphtheria toxin is a correct example, very correct example. Now coming back to this, please listen very carefully. Now diphtheria toxin is a protein, and you know why it is toxin? Because it inhibits a thing called elongation factor 2. Elongation factor 2 inhibitor is diphtheria toxin. Now what is elongation factor 2? So if you have learned translation process, how many of you have learned translation very thoroughly? Don't mind, I'm not taking your class, I'm just asking for the sake of learning only, nothing else. So what exactly happens during translation? One particular tRNA will be there. The tRNA will donate the amino acid on its head to a growing polypeptide chain. Now during this, what do you think, how this amino acid from the tRNA will transfer into a growing polypeptide chain and make a peptide bond? There must be some interfering factor, no? This is called elongation factor. So elongation factor is the factor, it's a protein which enhances the growth of the polypeptide chain. Okay, it literally works as a hand. It takes the amino acid from the tRNA and put it into the, so consider this like a thread, like you are making a garland. During the puja, what you do, you take a needle and then put all the flowers in between this, and then what becomes, it becomes a garland. Okay, Mary and all other those who are not from India, they maybe this thing. This is kind of a necklace. Okay, so in necklace, what happens? There are multiple pearls. So how do they put the pearls? You take a needle and then one after the another you you put up the pearls, because on the pearls there are some holes. I'm not sure very much whether the foreigners can understand what exactly I'm trying to say. So this elongation factor 2 helps in developing the polypeptide chain. So can you tell me what would happen if the elongation factor 2 is inhibited? Then protein synthesis will completely shut down. And do you know what exactly happens during diphtheria? The muscle will become a rigor mortis. Rigor mortis is the spasm of the muscle when a person dies. After death, no, if you keep the dead body just like that for a longer period of time, you will see that it is getting squeezed. The body is becoming smaller because of a mechanism called rigor mortis. Rigor mortis is nothing but a mechanism where all the calcium channels are opened in the muscle, and therefore, okay, in therefore, what exactly happens? All the it seems that the dead body is trying to sit down from the bed. Okay, I do not know whether you are able to like visualize the thing or not. So diphtheria toxin also does the same event of thing, and it's a highly toxic. So this is one. Now second one, cholera toxin. Have you heard of cholera toxin? You must have heard. So cholera also produces a toxin. Now, if I do not know whether any microbiology person is there or not. Now, who can tell me whether you have heard about anti-streptolysin O or streptolysin O? It is also a toxin. It destroys RBCs. Okay, it is produced by a bacteria called streptolysin bacteria, which is nothing but the streptococcus, which causes sore throat, and if it remains untreated, it will convert into a rheumatic disease, rheumatic heart disease, rheumatic fever, and all such things. Okay, so this is also a toxin. Then some more toxin, let us say, protein-based toxin. Then this diphtheria toxin, cholera toxin. Then one more very famous toxin is there. Okay, so that toxin now is useful for a this thing also, plant, genetically modified plant. That is, who can tell me which toxin I am talking about? Bacillus thuringiensis toxin. Okay, so it is toxin not for human but for bollworm. Bollworm is a bug which destroys all the this thing, brinjal crop. So to kill that brinjal crop, there is genetically modified brinjal. That is BT brinjal. Ah, you are right, BT toxin. Understood. So these are toxins. So what we'll do now, we'll search for a toxin. Okay. Now Shayak Mondal, what you do is you open this thing. Oh no, not Shayak Mondal. You what you do know, you this thing, open the this thing NCBI protein. Okay, NCBI protein. Fine. Now here we'll search for some toxins. So which toxin we shall search for? We shall search for streptolysin O. You write down streptolysin O. STREOP. Streptoto. Strepto. Strep. No. STREO is not there. Sorry. Strepto. STREP. P. T. O. Strepto. Streptolysin. Lysin. Lysin. Streptolysin. Then dash O. Dash O. Search. $571, then 195. So 195 faster. Second one. Okay, copy. Go there, paste. Very good. Very good. Run. One more time submit. Submit नहीं हुआ. No. So we'll see the methodology. Why? Okay. Shayok, go to the home button. Go to before back. Okay. Achha, Shayok, what it came show? Uh, can you share what exactly, Shayok, what you have got?
So here in the screen.
No, Shayok is saying that he has got a separate result. So I want to know what he has got. No, he's a participant. So okay. Oh, it came toxin, na? So what is the score? Acha, in the meantime, no, you go for prediction. Uh, okay. Good. Acha, go for prediction one more time. Okay. Now, no example sequence. Example, में click करो. So just hold on. Acha, now you do submit. Let us see what exactly happens. If it can identify the protein, what protein is this? Let us see. Oh, many are there. No. Acha, but non-toxin, toxin. So the second one is toxin. Come back. Acha, can you copy the second this thing sequence? Copy. Now you go to protein. That protein database was there, no? There you go. Here, no, in the window you paste it in the window directly. You delete it and then paste the sequence. Enter. Okay, fine, no problem. Here you go. Where blast. Okay, Shayok, you go back. One more time back. Okay, now run blast. Somewhere it will be shown. Run blast. Can you see where run blast is there? Run blast is there. It is not there. Okay, Shayok, you select. Yeah, here you click it. Fine, very good. Okay. Click run blast. Search window, no, delete it. BAN. This thing is delete. Paste it. The sequence you have this thing, no, paste it. Very good. Now go down, blast. Acha, okay. All participants, please understand what exactly you are trying to do. In this tool, they were having some examples. The second example was identified as a toxin. Okay. Now we want to know what toxin is this, because it is not giving us any idea. We must know on what basis this particular tool is giving us the result. To find it out, we have a method which is called this thing blast. Now blast is nothing but if you search any sequence with a blast, it will tell you what sequence it is basically. Okay, Shayok's result is very good. It's a toxin. No, it definitely must be toxin. Anyway, no problem. So what we'll do is use preserve this screenshot, Shayok. Okay, now let us see what exactly kind of this thing it can find it out. If it can find it out at all or not. Although it's a very small sequence, we just want to know what exactly. So if you have a sequence that you do not know from where it came, so what you do, you go to protein, NCBI protein, then search for blast. It usually will be there somewhere. Then you click on blast and then put your sequence there and go down and click on the blast tab. So what it will do is it will find out throughout the database. It will take a little time, but finally it will give you a this thing, this what you call result. Okay, it is also something like a pattern recognition, but very basic. It is not predicting. It will only show whatever is there. Are you getting my point? Can anybody tell me what is the difference between predicting and non-predicting? Predicting means it was not there. Like you are using Chat GPT. You are saying that okay, write a biography of any author. So what it will do? It will predict. It will see somewhere and then it will say. But in Google, if you say that write, show the biography of Professor G.M. Ramachandran. So it will show you Professor G.M. Ramachandran's original biography. So it is not synthesizing anything. Until and unless something is synthesized, it is not intelligence. It is only like in library. The librarian will be there. He do not know any book, nothing he knows. But he knows where the book is. So he will go somewhere, bring the book for you. Understood? So databases are something like a librarian. It does not synthesize a result. But whenever you get some result, like Shayak Mondal just shared, no, it's a toxin, then this is the probability, this is the this thing, efficiency, and then what else? So this is a prediction. This needs an algorithm. Are you getting? So it will take a little time. In the meantime, we'll go to the next one.
Sir, I have a question.
Go ahead.
So
Go ahead.
the softwares we are using in toxin thread, like how
Uh-huh.
good is it? Because for me it showed it was the same thing came as non-toxin, but when someone else did it came as toxin. So is there
Okay.
something
No.
wrong with the algorithm?
Okay. No, there is a great possibility that this particular this thing can go wrong. But since it is our very known toxin molecule, so there is no point that it identifies it as a toxin. Many a times, what happens, some caches are there. So in computer, so what it does is it uh, it goes along with the cache memory many a times, but not always. Many a times you will also see that you are trying to log into your this thing banking web server, then it is showing that you are already logged in, therefore you come back later on 1 hour late. So if you go to incognito mode, the same server will respond everywhere. So there is a point, but this is no point that streptomycin O has been identified as a non-toxin. So if it does this repeatedly, we will reject this from our list. Okay, so if this does this regularly and every now and then it identifies a perfect toxin as a non-toxic, then this tool is of no use. Your point is also correct, but I have not checked it out. Therefore, I'll not comment right away. So I'll see everything. So whatever, no significant similarity found. So again, I am not putting any comment here because we don't understand why exactly this thing is going on and why it has identified a particular small thread as a toxin. So for the time being, we will not anymore use this because there is a lot of discrepancy. Now this is how your research will go on. So you will identify a protein and you will study through multiple different tools. Okay, some tools, this particular tool was highly cited and it has been published in a very good journal. So if we see that many a times this thing gives us these wrong notions, so we'll communicate with the journal and say that editor, this thing is there, but their algorithm is not very robust. This is a good point you made. Now I have shared one more tool with you. Now you please click on that link. You forget about it. Oh no, I'll come back to this tool later. No, I'll come back this tool later. Before that, no, yeah, here also I'll come back a little later. So I have shared one more link. Have you seen in the chat box? You click on that link. We must use a more robust thing now. Yes, very good. So what happens? All of you, please listen carefully. Now this is a tool that has been developed by the genetics lab of Harvard University. Okay. So the connection is not secure. Continue with the site. Okay. So here what happens, you go through a little, you will find that this particular tool, acha, go to home now first. Go to home now, we'll read little about this particular tool, what is its function and all. Oh, okay, okay, go to about now. So when we click on about now, we'll come to know, this is for reading purpose, that this particular tool name of this is Polyfan. Now this Polyfan is based on an algorithm which is called naive Bayes algorithm. Let us see where it is. Probabilistic classifier method. Naive Bayes algorithm is a probabilistic classifier method. Probabilistic classifier. They have not mentioned naive Bayes here. Anyway, probabilistic classifier method is a machine learning algorithm. Can you see the second line? High quality multiple sequence alignment pipeline. Then second one, probabilistic classifier based on machine learning method. Okay, and optimized for high throughput analysis. Now the beauty of this tool, now what you do is you go to the main page where the tool was there. Go back, put it back. Okay. Now this tool is useful to make you understand two different things. One, in machine learning, that you have already know that there must be a database. So in this one, you can see whess.db. Can you see it? whess.db. Can you able to see it? Ekdam terminal. No, you click on whessdb. Here you read it out. What they have said. See here, whess.db provides quick access to pre-computed set of Polyfan 2 predictions for whole human exome sequence space. Whole human exome sequence space. Throughout the whatever sequencing has been done from human genome, all the proteins makes into a database. That database is used as a training set for this, which they have termed as WHESS database. It contains annotations for all single nucleotide non-synonymous codon changes enumerated at each CDS codon positions in the exons of 43,043 USCS known gene transcripts. So 43,043 numbers of gene transcripts are kept in this database. Now this is the training set. Now what we'll do is we'll use this database. Now again, one more time, you click on the home page. Okay, fine. Okay, now this is the home page. Now here what happens, you know, in this method or in this this thing, what we call algorithm, we will learn to design mutations and also learn what is the impact of the mutation. Are you getting? How to design a mutation and to analyze what is the impact of that mutation. Okay, I give you 2-3 examples. All of you are already aware, but I'm giving you 2-3 examples. So most of the cancers are due to the mutations in the multiple different genes. Most of these genes are called proto-oncogenes. So proto-oncogenes means what? These are the genes which stops cancers. Oncogene means cancer causing gene. Proto-oncogene means cancer stopping genes. Okay. So now what we do is we will identify a gene which is known as a proto-oncogene. Okay, now going back to the protein server, NCBI protein, you go back to that server. You go to the home. Can you able to write NCBI protein? Okay, back. Okay, fine. Now in the search box, you delete it. Streptolysin, no, you delete. Now here you write BRCA1. BRCA1. Enter. Now go to Google and write BRCA1 so that everyone can read what is BRCA1. Go to any Google. Oh, okay. Here only it is in written. Achha, anyway, no problem. BRCA1. Enter. Let us see in wiki. Wiki is not there. Wiki. Very good. Now see what? BRCA is a part of a complex that repairs double-stranded breakup in a DNA. Okay, so basically it's a DNA repair mechanism protein, and therefore I'm calling it as a proto-oncogene. So whenever it is mutated, it cannot perform its function, and it will lead to breast cancer. Okay, so now you can read this out in your free time. So now going back to the NCBI protein sequence. Go to the NCBI protein sequence. Okay, so here you find it out. Homo sapiens, 19 sequences are there. Okay, great. So where is that? Just hold on. Just hold on. BRCA. Okay. Copy. Acha, Shagnik, what you do is you click on this link in the chat box. I'm in the chat box here in Zoom. I'm sharing one more this thing here. Just hold on. Okay. Shagnik, you click on this link. I don't know where it was not found. In the chat box of this. Okay. So this is the protein I wanted. BRCA partial Homo sapiens. Now go to FASTA. Okay, now copy. No. Polyfen. New tool. Then I'll come back to those tools. Now paste. Very good. Okay. Acha. Now Shagnik, find out what is the sixth amino acid. Sixth amino acid. LTKDRM 1, 2, 3, 4, 5. What is that, Shagnik?
Sir, it's M.
M or N?
M.
M, no? Methionine. Okay, fine. So now go to AA1 and select M. Go. M. Click. Achha, now please listen carefully, all of us. So what we are trying to do is we have selected the BRCA1 protein sequence. The sixth amino acid is methionine. It's a very famous position. this gene, if the sixth amino acid is mutated, what exactly happens? You will see now. So we have selected the sixth amino acid. In position box, see there is one more small box, no? There you write 6. Here you write 6. Okay. Sixth amino acid, methionine. Who can tell me methionine is which kind of amino acid? Methionine is not very much polar, not very much non-polar. It's a large, bulky amino acid. Okay. Now we will substitute this methionine with a strongly positive amino acid. Okay, lysine. Achha, go to down. Slowly, one by one we will read. A is alanine. R is aspartic acid. Arginine. Okay. N is aspargine. D is aspartic acid. C is cysteine. E is glutamic acid. So just hold on. Okay. Glutamic acid, right? Okay. Click on this. Click on this E and then submit query. Highly negatively charged amino acid, glutamic acid. Achha, now no, you read it out. क्या बोलता है? Grid gateway interface. Can you see? So grid gateway interface is nothing but the interface for mainframe computer. So here this is a very good example of how you can use remote computers for your work. So Harvard University will not produce any tool which is very unstable like that one, toxin pred. Okay. So what they'll do is they'll connect their server to the mainframe computer of Harvard University, which is usually called grid gateway. And you can see the Harvard's this thing logo also. Now, this particular calculation, this particular calculation is being run by the grid computer of Harvard. You are not downloading the whole thing because it's a huge thing. Your computer will go. Why to use? So we'll use through the server. Then now what we'll do is no load is light. People are not using this much every day. Right now. Now go to the refresh button. Can you see the refresh button? The result will come. Achha, one more time results. One more time refresh. Achha, why the error? Let us see what is the error. View error. Let us see what is the error. Key at position. Oh, okay. Go back. Go back one more time. Back. No, home. Now you paste. No, you paste without that greater than. Here you paste. Delete the greater than sign. You have deleted. No. Now do the same thing. Sixth minus it. Now let us see one more time whether it is correct or not. 1, 2, 3, 4, 5. One more time. 1, 2, 3, 4, 5, 6. Methionine only. Again, 6, position 6. Position 6. Then go to the same thing, aspartic acid or glutamic acid. Click. Then submit. What has happened? Okay. And then one more time you put. At first you put an okay. What is the wrong? What is wrong with this? One more time. 1, 2, 3, 4. 1, 2, 3. 1, 2, 3, 4, 5. 1, 2, 3, 4, 5, 6. 6 amino acid is methionine only. What is the problem? Submit query one more time. Refresh. No. The same thing will be there. So, go to one more time. Back. Paste the sequence. Okay. Now, no, wait. Now there is E and the second line is there. No, you keep the second line in the first. Means are you getting? हाँ वहाँ ऊपर लेके जाओ ওটাকে ওই যে জায়গাটার মধ্যে হ্যাঁ হ্যাঁ এবার দাও দেখি now do the same sixth amino acid is methionine methionine and then put glutamic acid submit query refresh you go to that protein link BRCA1 no the link I have shared no right now wait let us see okay you select everything from the beginning copy then go to the tool back mm, back paste now just hold on position 6 methionine then glutamic acid submit refresh results okay now here you see this is the result okay so what is the this thing we did we changed methionine which is a less polar amino acid less polar to a completely highly polar aspartic acid sorry glutamic acid okay now what is the result now here you see the result is the mutation is predicted to be possibly damaging with a score of 0893 and the sensitivity is 082 specificity is 094 okay so this is very stable software so how do we check it this is stable Shagnik, go to again Google write Google Scholar write Google Scholar in the search box correct go to the page okay now go to the tool leave it here go to the tool okay again go to back okay just hold on about select the first reference select the first reference Abid Zubei copy then go to Google Scholar paste submit how many citations cited by 3,302 people till now okay let us see the latest citation click on this cited by okay let's see what is the year 2017 2022 is also there can you see that also in Nature reviews can you see so these are its citations so this gives you an indirect idea that how stable your software is okay this is an indirect method of doing this kind of work when you come across some difficulty. Now, if you do the same thing in toxin trade, I'm sure that it will not give you this many results or this many citations. Anyway, so what exactly we did is we used two tools till now. One tool responded well and another did not. Okay, now going to the next tool. So what was the idea? I'll go slow one more time. BRCA1 is a proto-oncogene, means it is helping not to get cancer. Now, when there is a mutation in the BRCA gene, that is BRCA protein. So what it will do, it will not able to work properly, and therefore, what would happen? It will give you, it will lead to a cancer, and most probably it will be a breast cancer. Okay, so BRCA1 is a protein which is responsible for breast cancer when it is mutated. So what is mutation? So during COVID, everyone came across mutations. So what is mutation? So mutation is a method through which the structure of a protein is changed. Okay, at first the change takes place in the DNA, it gets reflected in the protein. From the protein, it causes a disease. Okay, fine. Now going to the next one. Shagnik, go to the next tool. What we are having? You have already clicked them. Okay, now this is what? Job status, submission detail. Okay, CSM peptide. Okay, now this is a computational tool to predict medicinal peptide. Okay, therapeutic peptide. Okay, so therapeutic peptide means, who can give me an example? Who can give me an example of therapeutic peptide? Let us say that and not antimicrobial peptide. Therapeutic peptide could be like the peptide which helps for maturation of RBC. Okay, what is the peptide that is responsible for maturation of RBC? hmm? For example, the peptide which is responsible for hematopoiesis. Okay, so the proteins responsible for hematopoiesis are the proteins responsible for hematopoiesis are uh, mm, let us Google it. Open Google one more time. Here you write peptides for hematopoiesis. Peptides for hematopoiesis. HEMATOPOISIS. Wait, granulocyte, CFU-GM, granulocyte stimulating factor. What else? Some more should be there. Production of cytokines. No. Hematopoietic factor. Uh, uh, Shayok, you change the terminology. You write hematopoietic factors. Hematopoietic factors. Peptides for काट दो. Peptides for काट दो. हाँ, enter. Erythropoietin. This is the thing I wanted. Okay, go to that protein. Go to that NCBI protein again. Technique. Here, no problem. Here you write erythropoietin, ERYTHRO, erythropoietin. 18. Enter. Acha, back. Delete one by one letter. No, one by one letter. I think this thing is not the erythro. ERYTHRO. Delete. They'll prompt. Wait. They'll prompt. No, erythropoietin is not there. Erythropoietin. Uh, okay. Delete all this. You write EPO in capital letter. EPO. Homo sapien CPO. Homo sapien CPO. Now go to what is there? Contigs. Just
So you're not audible.
Sorry for the interruption. So what we are doing is we have found out a sequence which is called erythropoietin. Okay. Now erythropoietin is a kind of therapeutic protein. Why? You know, the
Ma'am, can
kidney
you
patients are
give me
there, no?
the permission to share?
Uh-huh. So the kidney patients are there. They cannot able to produce erythropoietin. So they should be given exterior erythropoietin. Okay. Because I do not know whether you know or not. Kidney is not only a this thing filtrating device, it is also having a significant what do you call excretory activity. Okay. For example, it produces aldosterone and then glucocorticoids, you know na. So along with that, it also produces a protein called erythropoietin. I'm sorry for the interruption that has been taken place. So now we are having the sequence for erythropoietin, I think. Okay, the second sequence. Now go to FASTA, copy it, then paste. You do the way you were till now doing, no? The same way. Copy the sequence and see what exactly happens. Okay, and then run prediction, right? Here, submit, that is right, or upload. Okay, ठीक है. Email is optional, then submit. Let us see. Very good. Let us see. Submit. So the idea was to show you how to reduce the cost by using artificial intelligence. Okay. And machine learning for biomedical research. That was the main goal. Wait. Back. Acha. Can you copy the whole EPO? We go to EPO. From beginning, you select. Copy. Then go. Paste. Then again submit. Okay, we are rejecting it again. Close it. Next tool. Okay. Acha, why? Okay. Can you put a back? Nothing comes now. Acha,
No,
just
sir,
hold
nothing
on.
is.
The home page, job queue, finished. Okay. Help news. Acha, can you able to click on the IQ deep? The logo is there, no? Click it. Okay. Now here, no, this particular tool is useful for to identify three things of a protein. Please understand. Three things of a protein. First and foremost, which is called the secondary structure of a protein. Okay. Who can tell me what is secondary structure? Secondary structure of a protein is very important. give you one example. Like hair protein is there, no keratin. Keratin is made up of only alpha helix. So alpha helix is a secondary structure of protein. Then we are having hemoglobin, no, it's a globular protein. It is mostly by and large made up of globulins, that is beta sheets. Okay, so this is why protein secondary structure is important. Now, second thing is surface accessible sites, which is also known as SSA. Surface accessible site is nothing but in a protein structure, which are the area which is exposed to water. Are you getting? Consider this. Please understand. Consider this. You have taken the egg white. Egg white is albumin into a into a glass where it is everywhere water. So now what is happening? Your albumin is surrounded by water, no? So now in albumin there are many proteins which are interior and some many amino acids which are interior or in the core. There are some amino acids which are on the surface. Are you getting? So the surface amino acids are very important because they only make bonds with the solvent. Okay, it's a little bit high level of protein molecular understanding. So again, on protein, there are some amino acids which are having solvent accessible surface. So these are more important because they directly connect with the solvent. Now in human body, the solvent is water, but it's very important that these amino acids make interaction with the solvent. Otherwise, catalysis will not take place, muscle contraction will not take place, many other things will not take place. So this server will give you, it's a very robust server, will give you multiple things, and the major two things are the secondary amino acid sequence or the secondary structure of a protein, and then the surface accessible residues. So yes, we'll go directly to the protein server. EPO, we'll select the EPO on EPO sequence only the sequence will do now. I think the sequence will do now. Let us see. Copy, go there, submit. Okay, then what is this red color? Invalid
Sir, it
PDB.
is saying invalid
Oh,
PDB.
acha, PDB. Okay, no problem, no problem. Just hold on. They want a PDB, no problem. Acha, have you downloaded one PDB till now? Have you downloaded any PDB? You
Yes,
have
I
not
think I
downloaded,
have
no?
a few.
But they have not seen how to download PDB, I think. In this class, I think I have not shown. So what you do, you go to RCSB PDB. So please listen, students, participants. The RCSB PDB is one more database from where we can download 3D structure of a protein, which has been deposited by X-ray crystallography. Okay. Very good. Go. Akanksha Mishra knows about it. Very good. Don't worry, I'll give a little example what is X-ray crystallography, why it is important. Achha, here no, the same protein. Can you write EPO? In the search box, you write EPO. EPO. Search. See what happens. Okay, now we'll check the resolution. Select X-ray crystallography. Come down, down, down. X-ray, X-ray, X-ray. X-ray. Very good. Then select resolution. 2222. Where resolution is where? 2 to 2.5. Very good. Now go to the triangle and close it. Let's see a small structure. Go down, go down, go down. A single molecule. Go down, go down, go down again. Wow. Go down, go down. This one will do. This one will do. 1D6P. Click on it, 1D6P. Then go to download PDB format. Has it been downloaded? Now here you upload it. Fine. Open. Wait. It has not yet been uploaded, it seems. Copy and paste a model or upload. We have uploaded. Now what? Customize job parameter. See, customize job parameter, scoring mode, individual, then scoring resolution. You're right. Standard, high. Okay, now then see run. Is it running? Okay, Shagnik, you copy this. You copy the web list. That means the web, this thing, not secure, Fusion CS. Can you see the top, the web address? Can you able to see the web address? Select it. Copy. Copy. Okay, now go to incognito. Incognito. Paste. Enter. Upload. Oh, job name is required, it seems. Arey yaar, shit. Oh, okay. Job name, EPO. Here you write EPO. What they have said? Wait. Invalid job name.
Invalid job name.
Acha, in job name there is a question mark. Click on it. See which kind of job name they want. No space, special characters allowed except 1 and 2. Length should be 4 to 10. Okay. EPO 1 to 3. You write EPO 1 to 3. Upload. Fine. Go. Run. Wait. What they have said? Your job. Click here. Okay. Here you see. Can you see? A little bit up. Scroll. Scroll down. Wait. Here you see sequence, secondary structure, and solvent accessibility. Can you see? So SS means secondary structure. Where you are seeing H is helix. C, coil. E, anything. Next. Go. Coil. Like this it is. Okay. It will go on. Acha, close. Go on. See H, C. Can you see? H means helix. Okay. You want to see what is helix? So go to PDB. Go to that PDB that you have already opened, no? Where is that? Here you see. Can you see what is helix? Can you see? Very less beta sheet. Can you see the green arrow? These are called beta sheets. Okay. Can you see the threads? These are called coils. Okay. So many of you already know But here what happens is you need a quantity. Quantity means what, you know, if you want to know what percentage is alpha helix, then from where, from this structure you cannot find, then you have to use that tool. Are you getting my point? Why you are using that particular tool? It is to quantifying the degree of secondary structure motifs. How many are alpha helix? How many are coil? By looking at it, you can find out, but you cannot write a number. You cannot say, okay, 65% or 75%. You cannot say, isn't it? So this tool will help you. Now, second one, solvent accessible site. Okay, go back to the tool. The result. Here, no, result. That result was there, no? You have calculated. Great. So here, what they have done is scroll down. Great, Chagni. So as you can see, E means what, you know, exposed. B means buried. Buried means it is interior. Okay, it will not do any kind of interaction with the solvent. Are you getting? So multiple more this thing analysis are also there, but these are the two very important analysis which this particular tool will do and help you out. Okay. Now Sagnik, going to the next tool. What is the next tool we have?
So this was the last tool.
Okay. So till now we have seen some good and some bad results. One very good result is to identify cancer or the possibility of a cancer by using probabilistic classifier tool, which is being built by Harvard University. So in that, what we did, we took one protein sequence, which one, BRCA1. When we used BRCA1 and mutated the sixth amino acid, then you could have asked me, sir, why you have selected the sixth amino acid? Because the sixth amino acid mutation is frequently seen in Angelina Jolie's maternal cell line, this thing. Okay, her mother is also having the sixth amino acid mutated. Her maternal aunt is also having the sixth amino acid mutated. Okay, she is also having the sixth amino acid mutated, but with different amino acids. I do not know, if you can able to correlate. So if you have a mutation there, no, there is a high possibility that you will end up with a what? Cancer. So you can spend 6,000 rupees and get your BRCA1, what do you call this thing, screened whether you have any mutation or not. The sequence analyzer will send you the sequence, and the mutation profiling you can do yourself, because the BRCA1, that sequence we shared with you, that is the unmutated, means a real version of the BRCA1. If anybody is there who is having no mutation in BRCA1, that is the sequence of that particular protein that we shared. I think I have shared it is there in the chat box also. I think I have shared one sequence. Shagnik was trying to find, but he could not find it. So I shared one sequence, which is the partial, means not the complete one. Despite of it, it is a very good sequence of BRCA1, which is unmutated version. Then we introduced a mutation from which we got what is the end result? End result is possibly damaging, means it could cause cancer. Why? Because BRCA1 is a protein which repairs the damaged DNA strands. Okay, then we have used one more tool, which was toxin pred, but there were some discrepancy, but we must have to check it out. Then one more tool was there, which was not very good at all. Then finally we came back to this particular tool, which gives us an idea about the secondary structure of a protein as well as the surface accessible residues. Okay, so while studying a particular thing, we need to drive our focus in such a way that we start with some particular topic. Okay, for example, if somebody want to study antibiotic resistance, then he or she should take a protein and analyze that protein using all these tools. Then he or she should get an insight, which kind of tool is this, which kind of protein is this, and multiple different aspects of it. I have also shared some of my research papers. You can also able to go through, and you can get some motivation how to eventually use these artificial intelligence tools for your future research. By saying so, I would thank all of you to remain attentive. And if you have some specific query, you can ask me. I'll be happy to answer. Okay. So, coordinator madam, if